In this video, we'll be talking about enzymes, and in specific, we will be talking about the structure and the function of enzymes. Okay, there's a video you can watch here. First of all, you can find that in the PowerPoint. So, enzymes are molecules that act as catalysts to speed up a biological reaction. Enzymes are not consumed during the biological reaction. And the compound on which an enzyme acts is known as a substrate. Okay, it's just some key points to start with. Enzymes can break a single structure into smaller components or join two or more substrate molecules together. Okay, this is the difference between a catabolic and anabolic reaction. All right, so there are lots of different types of enzymes we have in our body. There are lots in other organisms as well, but here are just some examples to name a few. So amylase, protease, lipase, bile. So hydrochloric acid and bile are not enzymes itself, but these are just other molecules found in our body. The amylase, protease, and lipase are all enzymes that we have, okay? We notice that the ending of the name, the suffix is "ase", okay? Ase, and ase. Usually enzymes end with "ase", the suffix "ase", and that's the easiest way we can identify enzymes. Okay, here are some examples of what some enzymes look like. This is just the protein structure of these enzymes. Okay, now the important thing to know is that enzymes are proteins. Okay, they're just folded proteins that are in their tertiary and quaternary structure that have been folded in a specific way to allow for molecules, substrates, to bind to those active sites. Okay, here are some examples of some enzymes and their role. All right, so we're just talking about active sites before and what they were. Okay, this is an image of an enzyme itself. Okay, and we'll start looking at what each one of these points do in their function. Okay, so the substrate molecule itself. So there are chemicals that an enzyme acts on, and they basically bind to this space here in an enzyme, okay, known as the active site. So the active site contains both binding and catalytic regions, okay. So this is going into here, allowing that reaction to take place. We've got the enzyme molecule itself. So the complexity of the active site is what makes each enzyme so specific. Okay, the active site of the enzyme is very specific to the substrate and the reaction that takes place. Okay, so here's just to recap. Enzymes have a specific region where the substrate binds to. Active site is usually a cleft or a pocket. And enzymes are substrate specific, allowing specificity varies from enzyme to enzyme.